In this video, we look at Higher Human Biology, Unit 2, Physiology and Health, Key Area 3, the biology of controlling fertility. So, firstly, we look at Key Area 2.3a, periods of fertility, explain the terms fertile period, why women are said to have cyclical fertility, whereas men have continuous fertility, and look at how the fertile period can be identified. Men are said to have continuous fertility due to the fact that sperm is constantly being produced in their testes. Women's fertility is said to be cyclical, as they have a cycle which leads to a fertile period of only a few days during each menstrual cycle. At that point, it is possible for them to become pregnant. Fertile period can be identified as a woman's body temperature will rise by around ½ degree Celsius after ovulation, and her cervical mucus will become thin and watery. So hopefully we know the term fertile period is the days within the menstrual cycle where a woman is capable of becoming pregnant. A woman's fertility is cyclical, as there's only a certain period within each cycle that she's capable of becoming pregnant. You can identify this using the temperature rise during the period after ovulation and the fact that cervical mucus will become thin and watery. Men's fertility is said to be continuous, as sperm are continuously produced in the testes after puberty. We then look at subsection B, treatments for infertility, looking at the following procedures and how they can treat infertility, so stimulation of ovulation, drugs that mimic the action of FSH and LH, artificial insemination, the in vitro fertilization, and intracytoplasmic sperm injection. Suggest then why pre-implantation genetic diagnosis is carried out during IVF. So, there are 4 key stages to in vitro fertilization. First, we stimulate ovulation. Ovulation is stimulated by drugs that prevent the negative feedback effect of estrogen on FSH secretion. Other ovulatory drugs can be used which mimic the action of FSH and LH. These drugs can cause superovulation. We then surgically remove the eggs from the ovaries. Eggs are mixed with sperm in a culture dish. The fertilized eggs are incubated until they have formed at least 8 cells. At this stage is when we would do pre-implantation genetic diagnosis, which we'll talk about in a second. Embryo transfer will then occur at the 8-cell stage, and the embryo is transferred to the uterus for implantation. In artificial insemination, several samples of semen are collected over a period of time. The semen is mixed together, different samples, and inserted into the uterus of the woman. This is especially useful when the male has a low sperm count. Artificial insemination is often carried out using the partner of the woman. However, if the partner is infertile or there is no partner, a donor can be used to provide the semen. Intracytoplasmic sperm injection, or ICSI. Now, ICSI is a form of IVF. IVF is just when the fertilization does not occur inside the woman. So IVF has ICSI within it. ICSI, the head of the sperm is drawn into a needle and injected directly into the egg to achieve fertilization. If mature sperm in the male are defective or there are a low number, ICSI can be used. So we talked before about pre-implantation genetic diagnosis. So pre-implantation genetic diagnosis can be used to identify single gene disorders and chromosomal abnormalities within the embryo. So if we have, say, 8 embryos which are fertilized in our culture dish, we would implant the embryos which do not carry these diseases. Some of the other issues with IVF that we can deal with are the drugs that cause the superovulation to allow scientists to gather the eggs for IVF can cause superovulation, so that if the couple are still engaging in sexual activities, the woman could become pregnant with multiple embryos and give birth to multiple living offspring. So we've discussed the following techniques: the stimulation of ovulation, drugs that mimic the action of SSH and LH as part of in vitro fertilization, which we've discussed also intracytoplasmic sperm injection being a part of. Artificial insemination being used when the male has a low sperm count, and if the male has such a low sperm count that he is infertile or sterile, we can use a donor. Pre-implantation genetic diagnosis is useful in identifying embryos which have chromosomal or single gene abnormalities and not implanting these embryos. The final section is the physical and chemical methods of contraception. We will look at examples of physical methods and chemical methods, and for each of these we'll describe how it is that conception is prevented. We first look at physical methods. The two examples on the left are the female and male form of sterilization procedures. Above we have tubal ligation, where the tubes, so the fallopian tubes or oviducts within the higher course that lead from the ovary to the part of the oviduct where sperm will enter for fertilization, are either tied or cut so that the ovules from the ovary and the sperm traveling up the uterus can never meet. So there's a physical prevention of them meeting. Similarly, if you go more proximal to sperm production, if you cut or tie the vas deferens, which is the sperm tube in the male, again, the sperm cannot leave the testes and exit through ejaculation of the penis, so cannot meet the female's ovum. We have the intrauterine device. So a coil is inserted so it crosses the cervix into the uterus. This irritates the lining of the uterus and damages sperm which try to enter the cervix. We then have barrier methods often used with a spermicidal. So sperm are prevented from entering the vagina through use of condoms or entering the uterus through use of femidoms or caps by an impermeable device. Those were the physical methods. We also have chemical methods of contraception. There's the oral contraceptive pill, which is a combined pill of both ovarian hormones, the estrogen and progesterone. These are injected into the female or taken in pill form, and they pass around the bloodstream to the brain. They cause the negative feedback effect that we talked about as part of the normal menstrual cycle, but because it is consistent and before a follicle has developed and ovulated, it prevents any follicles from developing or ovulating within the ovary. So it causes the inhibition of secretion of FSH and LH. The progesterone only or mini pill thickens the cervical mucus in the cervix. That's how the cervical mucus prevents sperm from entering the uterus and therefore not meeting the ovum. The final form of contraception is the emergency hormonal contraceptive pill. This delays or prevents ovulation. It should be taken somewhere between the 72 and 120 hour point after unprotected sex. As it delays ovulation or prevents it, the egg cannot meet a sperm in the oviduct, so fertilization cannot occur. Obviously, if fertilization has already occurred by this point, this method is ineffective. So we've described the methods of physical contraception, we've described the barrier, sterilization, and intrauterine device. We've talked about the oral contraceptive pill, mimicking the natural negative feedback response of the female, and the mini pill using a progesterone-only mechanism to thicken the cervical mucus. We've also talked about, in extremis, we can use the morning after pill, which prevents conception by delaying or preventing ovulation. Hopefully that makes sense. If you have any questions, please ask.